Our next presentation was really a panel discussion on sequencing and data sharing and consent. Understanding your genetic information will be a fantastic panel discussion. I'd like to introduce right now Peter Goodhand. He's the Executive Director of the Global Alliance for Genomics and Health, and he is also a member of the Global Genes Medical and Scientific Advisory Board. He is moderating our session today and will help introduce our panelists. So please help me welcome Peter Goodhand.
Thank you, everybody. And thank you to Nicole and the team for putting on the conference. And I think we saw this morning what a great start we got off today. And I hope we in this panel will be able to build on that morning session and set the scene for some of the deep exploration work that happens this afternoon. Same qualifier as Brad. I'm not a scientist. I have spent most of the last 25 years, though, trying to move health research either into products or practice or policy. And so this is a great place to be as we do that together. Matt and Brad today describe the potential and the promise of whole genome, whole exome sequencing. And our panelists will address that opportunity and the challenges in realizing that promise. We're going to cover a lot of ground in about 45 minutes and leave, hopefully, 15 minutes for Q&A. To keep this moving, the speakers will introduce themselves in the first 30 seconds of their 12-minute session. And we're going to get our warning note, right? I just wanted to kick it off by saying why this conference, the session, the group, is so important to the work that all of us individually and collectively are doing. My day job, as you just heard, is running the Global Alliance for Genomics and Health. We're young, 2 years old as an organization, and we had the great honor to be recognized last year by Global Genes for our work in collaborations in science. Today, we're over 350 organizations, 500 individuals, and in more than 50 countries. And that includes universities, research institutes, professional societies, disease and advocacy organizations, funders, and companies. About 40% are companies. But if there's an area where we need more voice, more input, it's patient advocacy. We're fortunate to have some great umbrella organizations, including Global Genes, Genetic Alliance UK. But we really need that voice. And there is no more passionate and committed agent for change in this world than the rare disease advocacy community. And often the potential for real disruptive change. So that's why we're here. That's why we've come to learn and to listen and to share with you some of our work. Our group is focused on creating the enabling technologies that make data sharing easier, faster, cheaper, and possible. We're also addressing some of the regulatory issues that are of concern and the many ethical issues that we've got to consider as we start to share more and more data. We're trying to catalyze real-life sharing, and that includes projects like our Beacon project, or most importantly for this group, the Matchmaker Exchange, and we can talk more about that later. But we have a core belief that if we have to work together to create those core conditions for the responsible sharing of genomic and health data. And even though this work I do was founded by research scientists, it was on the premise that it wasn't about bigger science, faster science, better science. It was about impacting human health. And that's why it's so important that we're grounded in the work that you do. The other thing is sense of urgency, sense of time. And I think you bring to us more than anyone else that sense of a timeline that has to be addressed. And we have to think on your terms, not on scientific terms or academic terms or business terms, on your terms. And I just want to share personally why that resonates so much with me. You'll see in my bio, I am a business guy who moved into this work. But personally, I had a 12-year experience as my young family dealt with the rarest of rare cancers. And we went through misdiagnosis, non-diagnosis, partial diagnosis, full diagnosis, and then some experimental treatments. And my children lost their mother at age 42, but they also lost their childhood in that process. And so for me, this is a very real, a very personal thing, because I remember that sense of urgency and that sense of powerlessness to deal with the unknown. And so I think with that as a personal note for me, great pleasure to introduce our three panelists. Our first presenter is going to take you through Sequencing 101, the basics of how you sequence on a large scale. Our second presenter will deal with the issues of consent and governance and who owns that data. And our final presenter will talk about making sense of the information. And so, Denise, if you will go first. I'm also really pleased to say that all three of these organizations are member of the Global Alliance I talked about. So it's a special pleasure to introduce all three of them to you. Denise.
All right, good afternoon. I'm Denise Perry. I'm a genetic counselor at the Illumina Clinical Services Laboratory, and it's part of my job to work on a clinical whole genome sequencing test for patients with rare and undiagnosed genetic conditions. So I'm here to talk to you about Sequencing 101, and I thought the best way to learn about sequencing is to take you through a real patient's case that came through our laboratory a couple months ago to learn about the process and how it might be beneficial to patients. So this is the story of RC. RC had signs and symptoms very early in life. In fact, problems were detected in pregnancy. And when she was born, she was unable to move on her own or breathe on her own. She had a significant NICU stay. But then she went through a 16-year journey to find an answer, where some of her symptoms included very low muscle tone and easy fatigue. She went to a muscular dystrophy camp and never had an answer for what she had. So this 16-year journey to find an answer involved multiple specialists across the country. It involved lots of clinical testing, including some genetic testing and multiple muscle biopsies, and not to mention time, travel, and money for the family. Now, some of her neurologists had suspected a genetic condition called congenital myasthenia gravis syndrome. We'll call it CMS. But they weren't certain if this was actually the diagnosis, and if it were CMS, what was the cause? Considering that RC had been through a 16-year journey to find an answer with nothing to be found, they asked, could whole genome sequencing provide an answer? So what is whole genome sequencing? In a nutshell, it's our technology to read each letter of our genetic code. Now, you may have heard of exome sequencing. Exome is the part of the genome that actually codes for the proteins. It provides the instructions for the proteins. The exome is only about 1% to 2% of the genome. Now, since we have the technology to sequence the whole genome, we're really limited in our understanding of the genome, and most of what we understand lives within the exome. But our understanding of the whole genome is changing over time. It has the ability to capture genetic variation beyond the exome, but we're really still learning about that genetic variation and what it might mean for patients. So how does it work? So it starts with the blood sample, or the lab extracts DNA from that blood sample, and we don't sequence the genome from start to finish. We actually chop the genome up into lots of different pieces. Each piece is sequenced multiple times, and then all of these fragments are aligned to make one big sequence, and they're aligned in reference to what's called the reference genome. And since each fragment is sequenced many, many, many times, we can be much more confident that each particular base is an accurate call because we've done it multiple times. And what we do is we look at the reference genome in comparison to the patient's genome, and we say, well, what's different? Now, that's the easy part. The hard part is, what do those changes mean when we find them? And so it's not apparently obvious when you see a change what it might mean for patients. We have to ask some questions. What gene is this change in? Do we know what this gene does? What does this change mean for this gene? Does it actually disrupt the function of that gene? Or is it benign? Is it just part of normal human genetic variation? Each of us has about 3 million variants in our genome. Not all of them cause disease. Or we might have absolutely no clue when we come across a variant. So that's why when we see a variant or any type of genetic variation, we really need evidence. We need to say, oh, we've seen this in patients with this set of symptoms, it must mean something. Or these cells have a different function than normal. When might this sequencing technology be beneficial? So it's really useful for individuals with a suspected genetic condition. And right now, we're really best at detecting things that are due to a single gene disorder. There's several advantages over single gene testing or panel testing. So for single gene or panel testing, you have to know which gene you want to test. But with exome sequencing or genome sequencing, you can simultaneously look at many, many genes. And there's also the ability to do family-based analysis. So you can look at the genome of the patient in comparison to his or her family members and see what's different between the two. Now, family had used up a lot of resources in their 16-year journey. Whole genome sequencing is very expensive, but all the stars aligned when global genes and syndromes without a name came together, and through the Illumina iHope program, we were able to donate a genome to RC's family. So RC's sample and her biological dad and her biological mom sample came through our lab, and we did what's called a trio test, where we test the patient and their parents, and we compare the genomes and look for differences between all the genomes. And we actually ask questions like, well, which variants or combination of variants were inherited or are brand new in RC that weren't inherited from either of her parents? And which ones of these variants could explain her disease? So as we were going through this process, we found a variant in a gene called CHRNB1. And Dad, at a particular position, had a C. And the reference genome also had a C. So we would expect there to be a C there. But RC's mom and RC actually had a T there. So that looked a little suspicious to us. We said, well, what is this gene? Well, it turns out this gene is one of 12 genes that's known to be involved in CMS, but it's a rare cause of this disorder and accounts for only 1% of all causes of CMS. And we also learned that it's most commonly inherited in an autosomal recessive manner. So we looked at this is looking interesting. So we looked at this gene further. We said, okay, well, if it's inherited in an autosomal recessive manner, maybe one variant came from mom, and maybe another variant came from dad, or maybe there's a new variant in RC. So we looked at this gene further, and we found that there's actually a big chunk of this gene that was missing, and the arrows are a little bit too far that way. They should be that way a little bit. So there was a big chunk of this gene missing. We verified this finding in a different lab that has deletion testing and found that it was an accurate finding. So now putting this all together, we say, okay, dad is missing a big chunk of this gene. Mom has a single base change that's part of this gene that we predict would disrupt the function of this gene. And RC has inherited both changes, and resultingly, we would suspect that she has no working copies of this important gene. So as the laboratory, it's our job to summarize these findings and report them back to the physician. The physician's job is to put these findings in the context of the patient and clinical characteristics to say, okay, is this the diagnosis? Is this the cause of the disease? And the physician looked at all these results and did his own research and agreed that this was indeed the diagnosis and these were indeed the findings that caused the disease. So knowing the exact cause of CMS, well, sometimes when you know the exact molecular cause of a disorder, there might be drugs that are already available that target the pathway in which that drug works. And in fact, there was already a treatment beginning for patients with CMS, and RC began that right away. She's now moving around a little bit more freely, has decreased pain, and for the first time in 16 years, she was able to close her mouth for the first time. She wrote a lovely letter to our laboratory and expressed how great it was to finally have an answer and to have a name that she can share with her peers. So what does this case illustrate about sequencing for rare and undiagnosed genetic disorders? Well, right now, the highest likelihood of success is when the patient has a suspected single gene disorder, and clinical geneticists can help figure that out. What's the likelihood that this is due to a single gene cause? We're still in the early stages of interpreting whole genome sequencing. We still have a lot to learn about what individual variants might mean for human health. When an answer is found, sometimes there are treatments available, or there's an avenue for research for treatments that target that exact disruption in a gene. And sometimes it just provides an explanation, and that's valuable in and of itself. Many times we find variants that we don't know what they mean. Are they benign? Or do they contribute or cause disease? And sometimes clinical testing is needed to actually say for sure. More research is needed. And of course, sequencing lots of people and sharing that data helps us to figure out, oh, these variants are actually more common in the population. They can't be causing rare disease. Or these variants are very rare and also in patients with a variety of different clinical features. So that can really help us understand and unlock the power of the genome. I'd like to thank my colleagues in the Illumina Clinical Services Lab, and a special shout out to Dr. Shamul Chowdhury, who solved the case, RC's family and clinicians who provided such great information that helped us analyze her results. Of course, Global Genes and Syndromes Without a Name, and Illumina's iHope program and leadership who support our work in undiagnosed disease. Thank you.
So thanks, Denise. We'll keep moving through the presenters. So the next topic is going to be in the area of owning the DNA, consent, and governance of it. And Meg is going to take us through that session. Thank you.
I go back. Hi, everyone. My name's Meg Door. I'm a genetic counselor as well, and I work for Sage Bionetworks, which is a genetic and genomics think tank based up in Seattle, Washington. And I'm here to talk to you a little bit about phenotype and about consent. So let's talk about phenotype for a minute. So phenotype is what you see. So the unsexy part of the genetic sequencing story. We just heard about all of the cool things that you can do with your genome. In order to fuel that research, we really need to know about the phenome. We need to know about the physical characteristics and features that go along with our genetic changes. Phenotype became a really powerful tool when this monk, Gregor Mendel, started playing around with peas in his Abbey's garden. And so about 150 years ago, he made some huge breakthroughs in our understanding of genetics just by doing experiments with pea plants, and helped us understand the power of the phenome. When we think about phenotypic variation, there's a lot. So it's not just what we can see, like our hair color or our eye color or our eye shape or our limb length or the length of our gait, but it also has to do with the way that we develop, the way that we process information, the way that we respond to drugs, even our family health history. So phenotype is really a very broad spectrum of features that make up the constellation of symptoms in a condition or make up who we are as individuals. Oftentimes, phenotype precedes our understanding of genotype, as we just heard with RC's story. Down syndrome, for example, was described first as a syndrome right at the same time that Gregor Mendel was doing his discovery work with peas. It wasn't until about 100 years later that we understood the genetic reason for Down syndrome. This picture, I don't know if you guys know about Maddie Stewart. She is a model who has Down syndrome who was at Fashion Week in New York 2 weeks ago, caused major sensation there. So just like typically developing people, people with Down syndrome have a really wide range of symptoms. So even though they all have some common characteristics, each individual, just like the twins in our last slide, each individual has their own manifestation of their condition. And so we have two really important things to think about when we think about phenotype. We have the phenotypic spectrum, if you want to think about it that way, and then the heterogeneity that goes within that spectrum. So the constellation of features that we associate with a syndrome, and then the differences in those symptoms in different individuals. And this data is very, very important, and this data is very, very valuable. In fact, not having information, granular information about our phenotypic variability and our phenotypic spectrum is one of the major blockades to making advances in understanding human health. So how do we collect that data? We do it imperfectly. You go to the doctor, you say, he hasn't been sleeping well. Well, what does that really mean? Has he been waking you up 3 times a night? Does he always have the same symptoms when he wakes up? Are there certain foods that he's eating before he goes to bed that are causing these changes? Is it a different developmental stage? So phenotyping is really one of our major challenges, just as much as genotyping is one of our major challenges right now. How can we do a better job? How can we get the information we need to drive discovery? So the people I work with now, Sage BioNetworks, which is a nonprofit, has partnered with Apple to create iPhone apps. These apps are to gather phenotypic information in real time, both in passive and active ways. So what we're doing right now is testing to see if the sensors, the native sensors within the iPhone itself, could be used to gather real-time, granular phenotypic data about participants. This is like so amazing, right? You're thinking to yourself, wow, that is so cool. I came here today because of this rare disease, and we don't know everything about the phenotype, and my child isn't like that other person's child, even though we both have the same disorder, we're pretty sure about that. How could this data be used? This would be so powerful. We could discover the genetic reason for our disease. We could figure out target therapies. We could develop drugs. We could try out new ways of treating this disorder. You should be excited right now. You should feel like, oh, wow, this is really good. I'm really glad I came. But we must be cautious. We must pause for a minute. We need to really think about this. If we put an iPhone on your child's wrist, or if we start picking up data from the iPhone that's in your pocket, we're talking about a lot of data. Even if these studies are being done by scientists and scientists are doing a really good job with this data, we're using accelerometer data, we're using gyroscope data, we're using geolocation data. Suddenly we're talking about a data set that could identify you as an individual or identify your loved one as an individual. Do we really want to be collecting this data? Do we really want to be sharing this data? The answer is yes. We do. Don't worry. We're all on the same page here. So yeah, you really do. You want to collect all this data because you want to give it to the smarty pants in this picture. This is the MIT hackathon, medical hackathon. You want those scientists figuring out the reason why your child is sick. You want it. You want to give them massive amounts of data. You want to give them as much data as humanly possible. You probably already have had the experience of wanting to help find something that will help fix the disease that brought you here today. So how do we do it? How do we do it responsibly? Traditionally, we do this using a process called informed consent. And I'm probably talking about something that you already know. You sit down with a clinician, they tell you about a study, you hear all of the information, you ask questions, and then you sign this massive document that's written in legalese that you do not read, and nobody actually gives you time to read it, and you don't really understand it, but you're signing it because you know the clinician, and the clinician knows you, and you're trusting the clinician to gather the data to help solve the problem, right? This is not a scalable way to solve our phenotypic lack of information. This isn't going to work. If we really want to gather data from your iPhone or from your watch or from your Fitbit, it's not going to work to have you sit down with an individual clinician. We just don't have the resources to be able to do that. So how do we build this to scale? How do we scale up a consent process so that we're still honoring the fact that we're doing an informed consent process, an iterative process between understanding and processing information so that you know what you're doing. You're not just like clicking, I agree, I agree, I agree, I just want to get into this thing. How do we honor that information and understanding cycle? So hopefully this animation works. Oh, go back, go back. Let's see, will it work? I'm hoping to show you our consent process. And I know that the tech guys in the back are really amazing at their jobs. Look at that, it worked. Oh my gosh, you guys are awesome. I love you. All right, so look at this. This is what we did. This is the consent process that we developed at Sage so that we could use the iPhone as a sensor so that people could enroll themselves in a study. They could consent themselves. So it's a patient-centric, self-navigated consent process where you get to find out more information. We use icons to represent large ideas. We reinforce those ideas with more information. We're trying to make this interactive so patients or participants touch the screen, they interact with the screen, there are visual icons, there are words to read, there are videos embedded in this process to try to get more sensory integration. As a former teacher, that's really important to me, so that people really are grasping what it is that they're doing. We then get to this slide, and I stopped here for a reason, the sharing options. So you're donating your data for research. We're doing a very specific research study. But as part of the consent, we ask you, do you want us to have your data, just ourselves? Or do you want to share your data with qualified researchers worldwide? What we found is that over 75% of the tens of thousands of people who have enrolled in Empower and Share the Journey have suggested or who have stated that they'd like to share their data with qualified researchers worldwide. Think about that. When participants are asked what they want to do is share. We want to give. We want our data to be used to solve questions not only about the disease that we care about, but the questions about human health. If there was a way that my rare disease community could benefit another rare disease community, we all benefit. This is amazingly powerful. You're probably thinking to yourself, some of you might know about PEER, Genetic Alliances sharing platform. Okay, so just to like head you off at the pass. ResearchKit, the Bridge project, which is what this is called at Sage, the Bridge project is a little bit different than PEER. So PEER is a data registry where patients decide what information they want to upload, and it's entirely patient-driven. Bridge, the studies that I'm talking about here, these ResearchKit app studies, are studies that are designed by scientists. So we're specific about what data we want to gather from you, but we allow the participant to say, okay, you can gather my data for that specific purpose, and also I want my data to be able to be used in other studies. So our primary purpose is as a research tool. Our secondary secret goal, if you want to think about it that way, is to get more data out there. More data, more phenotype, more better. So what we're trying to do through this process is strengthen the chain of trust that exists between researchers and participants. As a participant in research, you are in a powerful position. You should know who the researchers are who are accessing your data. They should know who you are. It's a reciprocal relationship of trust that you're building. I had to go back to the P's. We owe a lot to Gregor Mendel. But remember, as a member of a rare disease community, you have a tremendous amount of power. You have your phenotype. You can describe it. You can donate it. And you can power the research that really changes human health. Thank you.
And to complete this, cycle, David Glaser is going to talk about making sense of this enormous amount of information that will be generated by the two approaches you just saw. So David's going to make it all make sense.
High bar there. Thanks, Peter. I hope the camera folks don't mind if I might not be standing in one place the whole time. I'll try to stay in frame. So yeah, Peter's exactly right. My background, I'm at Google. I'm a software engineer. I've done software my whole life. A couple of years ago, started looking for opportunities to take what Google was good at, which is take large amounts of information and find the value in them and say, can we help in the growing amount of information in this world, in the world of life sciences in general? And if you remember back, Google's mission statement when Larry and Sergey first founded Google was organize the world's information and make it useful and accessible. Because if you don't have organized information, you just have a pile of data. And what we've heard a lot of this morning already is all these new tools to get us more data. Awesome, if we can take that data and make it useful and accessible. So what I'm going to do over the next 10, 12 minutes is sort of state that opportunity, state it briefly, because you've heard it from Brad, you've heard it from Matt, you've heard it from Denise and Meg, and then walk through a case study, walk through an example of some work that's ongoing that we're doing with the Autism Speaks Foundation, and talk about how they're doing, how they're taking information and making it useful. And then kind of end on a checklist of here's what it might take to generalize that. And then we have a breakout later this afternoon where Anthony Philippakis and I are going to go deeper into that last part. So you'll have an opportunity to go deeper on some of that. So what does it take to make information useful? And just sort of putting it all out in one set of steps, several of which we've talked about in different places. But here it is in kind of a timeline. First, and Brad said this well, you have to know what you're trying to accomplish. Right? And so have a goal, have an understanding. Once you have a goal, you then have to decide what data will help me reach that goal. And that's going to be a combination of from what people. Do you only want to look at people who have the disease you're interested in? Do you want to look at their families? Do you want to look at people who are unaffected? Do you want to look at similar people? You have to decide where do you want to get information. You have to figure out how are you going to collect the biological samples, the blood, the spit, whatever the fluids are. You have to figure out how are we going to gather what phenotype information using some of the tools that Meg talked about, possibly. And then you have the plumbing. How are we going to do this? What are we going to do with all this information? What's the infrastructure we're going to use? What software do we need? How are we going to address the needs of security? How are we going to build tools to take this data and make it useful to researchers? And then once you've built this, you then have to say, all right, we've created this wonderful resource. Let's tell the world about it. Let's tell the people who actually might be able to use it and make sure the right people can use it. So that's an overall general process. Let me walk through over the next 6 or 7 slides. How did Autism Speaks set out to do this? What are we doing with them? And I think this generalizes well to the things that we've been talking about all morning. Autism Speaks has this project they call the Missing Project. And Missing is trying to build a large database of information about autism, genotype and phenotype information. Their goal is their first checkpoint is to get 10,000 people, whole genome sequences from all of them. These are, following up on what Denise said, these are mostly trios and a few quads. So they're all families with autism. They all have a kid that they're sequencing who has autism symptoms, but they also are sequencing both parents and sometimes siblings. So they're building this database. There's there are 1,715 that are alive as of today. There's another 593 coming later this week, and there's more on the way to the 10,000 number. So this is the shape of the project. The reason, of course, in autism that studying the genome is very aligned with their mission and with their goals is this strong evidence, and has been for a while, of how implicated it is. It's not as biologically direct a connection as some of the rarer single disease conditions, but there's a very strong genetic link. So the more information we get, the more people hope to understand and pick it apart. They're gathering both the genomic information and on the affected kids' phenotype information. And in the case of the Autism Speaks project, they picked a very narrow and very deep set of phenotypic information that they wanted to gather. And it's deep behavioral characterization to say, how is this kid's demonstrated behavior different from that kid, different from that kid. So that's, that's what they've done. You put this all together, and they've now collected, the raw data. I don't think you talked about the size, Denise, but the raw data from one person, depending, is about 100 gigabytes. And then you multiply that by 10,000 people. That's a fair number of bytes. You distill it down to the list of the 3 million variants per person. Well, 3 million variants per person times 10,000 people. It's still a lot of information. There's a lot of stuff going on there. And they wanted to take all this information and say, how can we make it useful? They started by saying, well, how can we make it useful to people like Meg and Denise, to the genetic counselors and the genetic researchers who have questions they want to ask? And that people want to ask questions. These are very similar to the examples that Denise put up earlier. There's something interesting going on in someone I'm studying. I'm a researcher, and I want to see if there's anything in this library that Autism Speaks has built that's related. Have you found any people with the same kind of variation? And if so, what are their symptoms, and how similar or different are those from mine? You want to be able to speak that language, speak the language of, and I list some other examples here of the kinds of questions you might want to ask. So what Autism Speaks did is they built a custom research portal that wraps all the underlying data, and I'll talk about the architecture in a minute so you can see how it's built. But let's start with the people who are going to use it to answer questions. They built a portal that lets a researcher or a counselor go in and drop down and say, I want to search this database. I want to search across these 10,000 people. And in this case, I'm interested in the SHANK1 gene, which is known to be implicated in some cases of autism, and I'm looking for a particular kind of variation. So Denise showed us 2 kinds. One is just a single nucleotide changing, the C to the T typo. Another was a deletion. Some deletions actually cause the protein coding to get out of whack. That's a frame shift. So in this case, I'm saying, you got anyone in the database with those conditions? And then you get back a list, and in this case, there were 3 instances of people in the database with variants that matched those criteria, and then we can say, oh, well, that's interesting, let me drill in. You click on one of them, and I've redacted any information that could even be conceivably confidential here. I don't think it is, but I was erring on the side of caution. And you start to get down and look at the phenotyping information that had been gathered on that particular person. And then if you're actually curious to say, well, but what's really going on inside the DNA? Well, then you can click a little further and say, let me look at the variants. Let me look at them in context. Then you can go further and say, let me look at the raw reads and say, where did that come from? So that's what Autism Speaks wanted to do with this information, wrap it with a tool that was useful to researchers. And they're starting to get results early, but the data that they've been collecting for this missing database has already been a paper published in Nature. You can see the highlights there. What's exciting to me is, yeah, it's awesome that they're getting results. It's more awesome that the data behind these results is now available to any qualified researcher. If anyone here is a qualified researcher, there's a link you can go to and you can request access and you can look at this data. And more, because they've released more data since this paper was done that hasn't yet been published on, right? It hasn't yet been mined. So they're consciously taking an open science approach and saying, how can we build things in a way that not only helps our families and our community, but how can we build it in a way that maybe not on day one, but is setting some precedent and giving us some things to learn from. That's what you would do as a researcher. There's a website. I think the link's there. I'm not aiming for researchers here, just sort of giving you a sense of what would a researcher do. They would click on that link. They would read that description. They'd open up one of those forums. They'd explain why they are qualified. They'd send it off to Autism Speaks. Autism Speaks would say, cool, you're in. And then they would get to start using the tools that we talked about and any other tools that they want to use because the raw data is available also. I have, I think, 2 slides of technology here. So how is this all built? And I'll keep it at a high level, but sort of what are the puzzle pieces that got put together to provide that meaningful, organized, accessible information out of all of this information? Well, it's built on the same infrastructure that Google over the last 15 years has used to power search and power mail and power maps and power YouTube. Google has this great infrastructure to, as I said, organize information and find the value in it. Google's cloud platform is how we take all that infrastructure and make it available to the world. And what my team does is layers on top of all of those services, a genomics layer. So you can now take genomic information, load it into Google's cloud, and start to use some of the tools that we have available for other things, because we've taught Google, oh, that's what it means, that's what reads are, that's what variants are. And then Autism Speaks built their missing database, their missing portal on top of that. So that's the stack. And the point of that is to let researchers use any and all tools that they want. So in addition to the nice friendly UI that we showed you, they can also drop down. I'm not going to talk through this. This is just kind of the flow of information into the system. So what Autism Speaks is doing when they get the data is load it into the cloud, import it into the system where it's available via a clean way for programmers to get access to it, a clean API, and make it available to all the different types of people who might benefit from this information with appropriate tools to the way they work. I do want to take 2 slides briefly to talk about access control and talk about, as Meg said, it's really important that first that we ask people who donate the data, what do you want to do with it? And then second, that we respect it, right? There's a phrase that I always use when people say, well, how do you decide who gets at the data? And I say, it's your data. You tell me, right? When you take pictures on your vacation, who decides who can see them? Well, you do. They're your pictures. That's the mindset we take to pretty much everything we do when we're working with people's private information, whether it's my photos or my spreadsheets or my genome. And in this case, Autism Speaks has a consent agreement with the families who donated information, and Autism Speaks are the ones who decide what is a qualified researcher. So when a researcher fills out that form, Autism Speaks then says if they approve, they add their name to that list, that read access list. And then once they're added to the list, they get to use all the tools we talked about because it's all gated behind that one thing. One of the beautiful things about that model, we also have an unrelated project going on with very related technology with the National Cancer Institute, where we're taking some of the cancer information, TCGA for those who know, that the NCI has collected over the years and saying, let's make this available through the same platform with a different set of phenotypes and a different front end, but the same technology. And if a researcher wants to access the TCGA data in the cloud, they ask dbGaP, they ask the NIH, may I? And the NIH says, yes, you may, and puts you on that list. And if you are a researcher who's on both lists, well, great. Now you get to cross-analyze. You get to look at all this information together and look for patterns. The vision that many of the people here have helped shape is we want to open this up to more and more collections of information that become, back to Peter's original, what's the purpose of the Global Alliance? We want to open up so that we really are sharing all the information with all the right people according to the consent of the people who granted it. So that's what we talked about as the steps. There's kind of a recap of the checklist. The parts in green are the parts that are pretty reusable that Anthony and I will be talking more about in the breakout later today and sort of walking through what would that mean. So I think I've run a little bit long, but thank you.
I'm swinging my microphone. So we have time. I don't know how much you want to stick to get us back. We do have 15 minutes. That's great. So and we'd like to hear from you rather than do more talking. But while people were coming to microphones or just doing that, I was going to ask each of the panelists, thinking about the room we're in, the context we're in, working with the rare disease community, both from the perspective of the 4 of us on the stage, but more importantly, working together with the people in the room. What's the single biggest thing you think we should be doing to really make a difference? So I don't know if I'm going to put you on the spot, Denise, since you're there. You have the most time to relax. So what would you say is the best thing we can do together? The other two get more time to think about it.
I don't know if my mic is on. So I think the single biggest thing is very basic. Everybody working together to create solutions for data sharing, collaboration, putting our heads together to solve some of these challenges in managing all of this data and figuring out what we as a community want to do with it and how to have it accessed in ways that are responsible, but also lead to better healthcare solutions and treatments and understanding science better.
And Meg?
I think, am I on? I think from my perspective, it's a point that came up earlier today. I agree very much with Denise, but I think we also need to be very thoughtful and deliberate. So I'm not saying go slowly, but I'm saying go thoughtfully. Just because we can do it doesn't mean that it's the best thing to do. So given all of the possibility that exists, just like when you're in the grocery store in the cereal aisle, like you don't want Fruit Loops, you don't want the Cocoa Pebbles, they're gonna give you a tummy ache, you know that. So you want to be thoughtful and deliberate in your choice. You want to be thoughtful and deliberate in your approach to all of this data, in the research that you fund, in the ideas that you forward, and broadcast more broadly within your communities. So I would say taking the time to be thoughtful and deliberate is one of the really critical pieces of advice I'd give.
And David, last word to you, and then I think we're ready for an audience question.
Yeah, I think I'm going to half agree and half vehemently disagree
Yeah,
with what you said.
I know.
Right, which is be very thoughtful about where you choose to focus your energy. We have cool technology between us, right? It's shiny, right? Ooh, shiny, right? Don't do that, right? Understand whether these are the right tools for your condition. I think both Brad and Matt had terrific checklists of things, when you have a limited set of resources, whether it's your time, your money, or both, decide where you think the right place to focus is.
Yeah.
And then, where I would disagree is that once you've decided, don't go slow, and don't let anyone tell you, no, that's not how it's done, or no, those aren't the rules, right? I think
Yeah,
the work
I
you've
agree
done
with
with
you
the work
there.
you've done, I knew you would,
Yeah.
but the work you all are doing with ResearchKit is a very different model that's got many fewer barriers, right? The work we're doing with Autism Speaks, they're working with their IRB to say, well, no, actually, our families really kind of do want this information to be useful. That's why they donated it, right? Don't tell me that's not how it's right. So I think you all are some of the best advocates for saying, do the right thing with our data once you've decided what the right thing is.
So I'll start the questions from the floor.
Is that on? Okay. As someone who runs a very small nonprofit, can you give me a ballpark estimate on how much this would actually cost? Because we're really at the point now where in the condition that I represent that we need to have a patient database and we need to start doing a lot of genetics and genomics analysis because it's something that has a lot of phenotypic heterogeneity. We want to work globally, and I just need to know what I need to think about in terms of funding.
Well, my short answer is once you've paid Denise, you're all set. The rest is easy. But that's roughly right. And I'm going to let you speak to where the state of the art is and what's possible on that part of it. But when you do a budget for something that includes whole genome sequencing as part of the project, the lion's share.
That's the part I know. That would be individual researchers. That would be the responsibility of the individual researchers, but to actually then work as the kind of center globally to organize all of this data, that's what I'm talking about.
Yeah. Okay, so check. They've already spent the big money. Obviously, the answer has to be it depends. But let me give you one answer that's both a little too big and a little too small, but it's a tangible one. If it costs about $2,500 to get a whole genome sequence done, and maybe it's $1,000, maybe by the time you conclude collection and shipping and handling and CLIA or not, but call it $2,500. Today it costs about $25 a year to store that data, in today's cloud pricing, and actually with the product that's coming out of approximately now, cut that in half. So for about $10 a year, you can store that up, call it $2,000 worth of data. That's not the whole thing at all, but it really is why I say that once you've got the sequencing covered, the bulk of the data isn't the hard part. There's big question marks around how much work do you want to do to build a custom front end? How much work do you want to do to organize the phenotype data and collect it, right? All of those can range from, we're just going to have everyone fill out a spreadsheet with 12 columns and load it into a table. Easy. To some of the things that Brad showed earlier with these really cool, interesting progressions over time of AT symptoms on different axes. That's a project in and of itself. And we should talk more, maybe Anthony and I will be talking about what we think it might take to make this real for different conditions, and we can drill into some of those it depends dimensions.
Thank you.
I would also add that one of the reasons we've put so much emphasis for the last few years on interoperability is so that we can build these tools once and they can be used many times. And if it's a slightly different tool, but you've used common underlying standards or APIs, then we don't have to reinvent the entire stack every time. And I think it's when you start to see a Google, an Amazon, and a Microsoft collaborating, or leading research institutes around the world, there's a real passion to let's make some infrastructure investments in core technologies that then everybody can benefit for the future.
So actually, Peter, I should have said that when I had my slide up. I'll be brief on this because I want to hear all of you. But when I popped up that Genome browser there, that was not a Genome browser that Google or Autism Speaks wrote. That was IGV, Genome browser developed at the Broad, one of the standard ones. The people at IGV, Jim Robinson, the developer, adapted it to say, oh, I will call Global Alliance APIs to get information. And IGV works with any data that's been published by the Global Alliance APIs. And Google, when I showed API, we used the Global Alliance APIs. So it just Lego, plug them together because of the work the Global Alliance has done to allow there to be one plug.
So we have a question that came in by Twitter that's up on your screen. So how can the MSSNG, the Missing Project, be scaled up and down for individual or the collective?
I'd say come to the breakout session because Anthony and I are exploring exactly that. We don't know the answers, but we have some thoughts.
So we hope the tweeter is in the room.
I know. I know.
But I actually think we answered some of those things. This idea of scale and the very expensive pieces may not be the genomic sequence and the analysis. It may actually be in the phenotypic side of things. So.
Right. Hello, Jeff Wichich, Cure AHC. My question has to do, we talked about your example on diagnostic. What about, we're at the stage now where we're looking at doing a natural history of our disease to prepare, hopefully, for clinical trials. And I'd like to get your ideas on, because we're looking at writing our own apps and things like that, the usefulness of this in that whole process of providing a natural history to go towards clinical trials and those sorts of areas.
Meg, that sounds like we start with you and then...
Yeah. The Bridge server, the Bridge project is available for collaboration. I just want to make that clear to all of you. We are a nonprofit. We would be happy to collaborate with other nonprofits who have research questions. So if you were trying, for example, to put some data behind the natural history of a disease, that's something. And it's something that sensors on a phone could help with, so passive data collection could help with, in addition to active data collection, like answering survey questions or doing tapping tasks, for example. I would be happy to talk with you more about that. But we really, the Bridge project is really designed to try to build up these large phenotypic warehouses to help drive research questions like yours. The data can then be manipulated on Sage's quantitative analysis platform, Synapse, which is a free resource that's available to any researcher to allow for large data manipulation. So, it's a little plug.
Hi, Tom Christie from Polyneuritis Foundation. What do you feel is the possibility of incorporation of all the legacy data that's out there now into a new warehouse or data marts that where you might be able to incorporate that with new things such as genome sequencing, things of that nature? Do you find any value in that?
I've got a partial, but go ahead.
There's tons of systems of record out there through healthcare.
There's tons of systems. Yeah. I think, integrating a lot of existing data into one is a challenging question in how we use it. And sometimes when we look at data, just if we examine the literature for different patient data, we find some important pieces are missing that weren't collected. No one thought to collect a certain piece. So I think, I'm not sure if I'm answering your question, but I think there's definitely possibilities, but there's also challenges in what do we know about that data, what was missing, and if it's missing, how do we collect it?
I think you'd agree that, patient history is an important part
Yeah.
of any analytics that you're going to do.
Yeah.
And, a lot of programs, I'm involved in one where we have to start from scratch because we don't have the ability to use that legacy data. But I just wondered if there was anything in plans to, be able to incorporate that.
Yeah,
Yeah.
so Meg's done a lot of work on all the many family history options out there, so I'll
Yeah.
pass to her.
So, I often say that we're in the grand adolescence of technology and health intersecting right now. And just like real adolescence is ugly and spectacular at the same time, so is our time in history. So we have lots and lots of data sources that are ugly in the ways that they collect the data and share the data, but are amazing in the richness of the data that they have. I think it's our challenge as scientists and as engineers to figure out ways of pulling those data together and also creating standards so that moving forward, we're collecting data in ways that are interoperable. And so there are a lot of organizations across the US and around the world who are working very hard in this space. The Global Alliance is one of them. The HL7 group is another one. It's awkward, it's ugly, but just like Chelsea Clinton, we're going to be gorgeous when we get to be older, right?
So
You remember, we remember. And she's so beautiful now. And the baby, oh my gosh, she's so beautiful.
all right. So we have one more question coming from the audience, but just before we do, so 2 comments on that. One is, I think Brad and others have often reminded us that the vast majority of the genomic information we'll be dealing with in the future doesn't exist yet. So we've come, so there is a lot of legacy data, but we shouldn't worry too much about finding all that legacy genomic data because we've got enormous capacity now to start and get it right from the beginning. The challenge is bringing in that rich legacy data on the phenotypic side or finding ways to interface differently with electronic health records so that we can get the advantage of both research and clinical care instead of seeing them as 2 solitudes. So one more question.
Hi, I'm Anne from the Atypical HUS Foundation, and my question is, we have a university team of researchers that are studying our disease. They do specific gene testing and whatnot, and so they're collecting the data, but as our foundation doesn't have complete control or access to it, and we're trying to look more globally with other teams of researchers across the world, how can we either merge their data into something like this, or is it better to start fresh off with a new database?
So I will say I'm going to ask David, I think, first of all, to talk about that because one of the really interesting things, I don't know if you saw it, but the Autism Speak is a US-based patient group, a Canadian academic medical center, and a global technology provider. So that's a good example of not having it done in your local state or province.
Yeah,
Meg,
I just want to jump in quickly and make a quick point, which is that you should require data sharing as part of any grant that you give. So make sure you write that into the conditions of your grant. If you're giving people money, they have to give back the data.
Yeah, and that won't be an easy conversation in all cases. You're in charge.
You've got the money.
So I think, and in terms of actually using the data, that's probably not going to be the hardest part once you get permission. It'll probably be, at least on the genetics side, easy enough to homogenize. Getting the permission is locking it up front. I'll give a tiny example of that. I know I'm over time. Not quite on the genetic side, but that UI that I showed that Autism Speaks had built, they hired a technology team to build that UI on top of the API. And the technology team said, oh, great, we're going to build it and we're going to keep the rights to the software so we can use it again. And Autism Speaks said, no. It's going to be open source so that anybody can use it for anything. And so it's open source because they were writing the check.
I'll just say one more thing too. Global Genes actually worked with Sage Bionetworks on a toolkit that is now available. We have some here, and it's online on consent. The questions to ask. We want to try to really boil it down to make it easy because a lot of times all this stuff is a little overwhelming and crazy. So thank you very much, and we'll have those available for y'all as well. But thanks.
So
to all
just
of you.
one concluding comment. It might not all, having been in your shoes in many ways, it may not always feel like it, but you have enormous power. And if you can exercise that voice individually, as groups, and collectively, you can actually cause more change and more disruptive change to the traditional ways of doing things than almost any other group that we deal with. So thank you.